In this video, I am going to be talking about the GPCR pathway. So what is a GPCR? Well, a G protein coupled receptor, commonly called a GPCR, is one of the most important signal receiving systems in the entire human body. They can detect signaling molecules such as hormones, neurotransmitters, and even odors, light, or taste molecules. But we will be focusing on the hormones norepinephrine and epinephrine. GPCRs are responsible for transmitting these signals from the outside of the cell to the inside of the cell in order to regulate a cell's activity. These GPCRs can be found all across the body in places such as the heart, brain, lungs, and eyes. The GPCR receptor itself is made up out of 7 transmembrane helices. The outside part binds to the ligand, the signaling molecule, and the inside part in the cytosolic side interacts with something called a G protein, but we will get to that later. So, as I've said before, the ligands I will be talking about today will be epinephrine or norepinephrine. These will bind to the GPCR's extracellular domain and cause the receptor to make a conformational change. On the cytosolic side of the GPCR receptor is the G protein. It is important to note that the G protein is not attached to the receptor until the ligand binds to the receptor. But the G protein itself is made up of 3 parts: the alpha subunit, the beta subunit, and the gamma subunit. The beta and gamma subunits are almost always stuck together, while the alpha subunit has a molecule attached to it called GDP. Whenever the receptor makes the conformational change, the alpha part of the G protein will switch its GDP for GTP, which frees the alpha subunit from the beta and gamma subunits. So, after the G protein has split off from the beta gamma subunit, it will go and activate either adenylyl cyclase or phospholipase C, depending on which kind of G protein it is. The G alpha S protein will go activate adenylyl cyclase, and the G alpha Q protein will go activate phospholipase C. So, but for right now, we'll stick to the G alpha S protein. So, after the G alpha S protein activates the adenylyl cyclase, that adenylyl cyclase, which is an enzyme, you can tell by the ending in ACE, that will pump out cyclic AMP, which is the secondary messenger. Next, cyclic AMP then goes to the regulatory units of pKa, and it'll bind to the regulatory units, about 4 cyclic AMPs for all the regulatory units, and then that will free up the catalytic subunits of pKa, which are kinases, which means that they are actually going out and phosphorylating target proteins. So now going back to the G alpha Q protein, after it activates phospholipase C goes and cuts PIP2 and into IP3 and DAG. IP3 will go to the endoplasmic reticulum and cause it to release calcium. This calcium is then taken up by calmodulin, which causes calmodulin to have a conformational change. Calmodulin then goes on to help activate PKC. And since DAG is hydrophobic. It can hang out in the cell membrane, and DAG will actually just directly activate PKC with the help of calmodulin, and that causes it to go out and finally phosphorylate target proteins.